Okay, hello everyone, welcome back to this channel. Today in this video, we'll be talking about transposable elements or the dynamic genome. So we will be starting with the question itself, what are transposable elements? Transposable elements are the segments in the DNA which are able to move from one place to the another. That is, these sequences, transposable elements are mobile sequences present in organisms. These transposable elements are also called as insertional sequences or K-sets or even mobile DNA elements. These transposable elements are found in all organisms, that is, in both prokaryotes and eukaryotes. Okay, prokaryotes plus eukaryotes. These organisms have transposable elements present in their genome, and they make a significant amount of the genes of the genome, I should say. So, in case of humans, if you see, 45% of the genome is composed of transposable elements, while as maize has 80% of the genome as transposable elements. Some people call transposable elements as simply transposons, but transposable elements is an appropriate term here. And the process by which these transposable elements move from one place to the another place is called as transposition. Yes, it's very simple, and they are also referred to as selfish DNA. That is, these transposable elements do not have any specific function for an organism in which they live. However, recent studies have suggested that these transposable elements are important in forming mutations, in important in evolution purposes. And it was due to this reason, because of lack of any specific function, they are also referred to as defective selfish DNA or simply as junk DNA. That means they are without any purpose. Okay. Transposable elements were first discovered by Barbara McClintock in 1983. He got Nobel Prize for their discovery. Okay, he discovered them in maize. As you can see, her photograph holding maize. If you look at this maize, there are variations. Any maize you look at, there are variations in the coloration of the grains. So she found that this variation is actually due to mobile genetic elements, which she called them as DS or disruptors. And these DS or the disruptors are actually transposable elements, which are activated by another transposable element called as activated by AC. And this system was then referred to as ACDS system. AC means activator of disruptor. Okay. Uh, these, as I said, these transposable elements are found in both prokaryotes as well as eukaryotes, and we'll be discussing different kinds of transposable elements both in prokaryotes as well as eukaryotes. First, we will look at the transposable elements in prokaryotes. So, in case of prokaryotes, we have insertional sequences. Okay, let me choose a different color because it is already red. So, insertional sequences are simply IS. You have composite transposons and you also have non-composite transposons. So we'll be discussing them in detail. First, we will look at insertional sequences. If you look at insertional sequences, these are about 1 to 2 kilobase pairs long. Okay. Now I should I should choose red color here. Okay. So these insertional sequences are about 1 to 2 kilobase pairs long, and they are flanked by IRs, inverted repeats. I hope you are aware about direct repeats and inverted repeats. Direct repeats is like if you have a sequence of DNA where you have ACGT, this will be repeated as such in the same DNA strand as ACGT. Okay, this is a direct repeat. However, if you have ACGT, and in the complementary strand, you will be having the same sequence but in reverse direction. Okay, ACGT. So this direction and this sequence runs from 5 prime to 3 prime from this direction, and this sequence runs from 5 prime to 3 prime in this direction. So these are inverted repeats. Okay, and in case of this, you have an inverted repeat. For example, if it is like A C G T C here, it will be C G C T. Okay, C T G CA here. So these are inverted repeats. And these insertional sequences are autonomous as they have the gene for transposase enzyme, which is involved in the transposition of these insertional sequences. And there is the signature of transposable elements or the insertional sequences in the target DNA, which is called direct repeat. We will look at how these direct repeats are generated. Okay, so this is about insertional sequences which are autonomous, have a transposase gene. So this slide shows how these insertional sequences move in bacteria. If you look at this very part of the diagram, you have this is an insertional sequence element. Okay, it is flanked by, as I said, IRs or terminal inverted repeats, and these inverted repeats are recognized by transposase, two subunits of transposase gene, and then these transposase enzyme subunits cut this at the TIRs. And then, as you can see here, cutting occurs here at the TIRs. And then this donor DNA, this is the donor DNA because it contributes the insertional sequence or the transposable element. So this donor DNA, okay, has this footprint of TSDs. TSDs means target site duplications. What does it mean? It means that the transposable element was present here and then it has been excised. Okay. Then it has been excited and that transposable element has moved to a different place. Now, one is this transposase enzyme excises this insertional sequence at that inverted repeats. It then recognizes this target sequence and makes cuts at the target sequence in a staggered way that we will look at in the next slide. And this transposable element gets inserted there and also forms these target site duplications. So how these target site duplications are formed, we will look at here. Let me take a different color here also. Okay. So these are inverted repeats. This is okay. These are inverted repeats. This is the example how these short direct repeats or the target site duplications get generated by the transposition. So if you look at this, transposase comes. Transposase first excises the these is elements at the inverted repeats here. Okay, and then this transposase makes a staggered cut. This is the target DNA, this blue color, this is the target DNA, and this transposase makes a staggered cut, as you can see. Okay, like this, which leaves TCGAT overhang in one side of the cut and AGCTA in the other side of the cut. Okay, as you can see here. Okay, here. These gaps are created by the staggered cut, and once this insertional sequence is inserted, these gaps are then filled by polymerase and sealed by DNA ligase, and hence these duplications are generated in the host DNA or the target DNA. Okay, which are called as target site duplications. Target site duplications are simply TSDs. That means these TSDs serve as a footprint that some at some time there have been this insertional sequence present at that very point. Okay, so this is about insertional sequences. Second type of transposable element in prokaryotes in bacteria is composite transposons. Composite transposons. What are these composite transposons? These are transposable elements which are flanked by insertional sequences. You have insertional sequence here, you have insertional sequence here. These insertional sequences have that we already saw that these insertional sequences have transposase gene, and these insertional sequences flank this antibiotic resistance gene. Okay, antibiotic resistance gene. So composite transposon has transposase gene. Okay, plus it has antibiotic resistance gene. Antibiotic resistance gene. And these are obviously they will be longer than is elementals because it has two insertional sequences at the end. Uh, they contain transposase and antibiotic resistance. For example, if you take of composite transposons, there is tn9 and tn10 in bacteria. Okay, tn9 has chloramphenicol resistance gene, while as tn10 has tetracycline resistance gene in them. And these composite transposons are believed to be medically important as these carry these antibiotic resistance genes, which help bacteria to get resistant against antibiotics. Okay, so third type of transposable element in bacteria is non-composite transposons. What are these non-composite transposons? These are sequences which contain antibiotic resistance gene but don't have flanking insertional sequences. They have terminal inverted repeats on both ends, which are recognized by transposase and help in transposition. And they also contain transposase gene plus resolvase gene. Okay, transposase, antibiotic resistance, and resolvase gene. Three genes they have. They also cause target site duplications upon transposition. And the important example in non-composite transposons is tn3 in bacteria. So if you look at tn3, it is 4,957 base pairs long. It has, as we already saw, it has gene for transposase, resolvase, and also beta lactamase or antibiotic resistance. This beta lactamase is antibiotic resistance. Okay, and these transposable elements, these tn3s or non-composite transposons, they use co-integration mechanism. Co-integration mechanism while transposing from plasmid to chromosome and vice versa. Because if you look at bacteria, suppose this is the bacteria, it has, let me change color. It has, okay, it has a chromosomal DNA, which is also circular, and it also has plasmid DNA. Okay, small plasmid DNA, spherical. And if this transposable element is present on chromosome, it will be transposed to plasmid, or if it is present on plasmid, it will be transposed to chromosome. Okay, this is what we mean by here, from plasmid to chromosome and vice versa. Okay, so how this co-integration mechanism takes place, let's have a look here. I hope it's pretty clear. So look here, there is this donor DNA in the blue color, and there is this recipient DNA in the blue color. If you look carefully, here we have arrows over here, and another arrow is over here. These arrows indicate the sites at which nicking by transposase takes place. Okay, as I said, transposase recognizes the inverted repeats. Okay, and nicks them. Then what happens? It also nicks the target sequences. Okay, it also nicks the target sequences, and then once nicking takes place, then there is fusion of donor DNA and recipient DNA with each other, and this kind of structure forms. Okay, and it this kind of structure has gaps at the in these transposable elements. It will have one transposable element strand over here and one over here. Okay, at that, at the junction of fusion. Okay, and then these transposable element gaps are filled by, okay, these will be filled by DNA replication. Okay, as you can see here, new DNA is formed as shown in purple, and then recombination takes place. One is replication takes place, replication or the gap filling takes place. Then there is DNA recombination taking place, because these two are homologous copies. These two regions are homologous copies, and once recombination takes place, as you know that there is rec BCD is involved. Rec A is involved. Rec A, which is usually required for strand innovation and branch migration as well to some extent. Then you have uh, RUVC, RUVC resolvase, which is a resolvase. Okay, which is involved in resolution of the holiday junction. Okay, once strand inversion takes place, there is formation of holiday junction, which is resolved by resolvase, and you get two. Okay, you get two. One is donor DNA and the acceptor DNA, both will be having a copy of transposable elements. Okay, so this type of mechanism is called as replicative transposition. Replicative transposition mechanism. Why? Because the donor DNA copies the transposable element into the recipient DNA. Okay, and there is the another kind of mechanism which is contrasted to this, that is called non-replicative. That is called a non-replicative, that we already saw in case of composed insertion sequences, where the, okay, let me go back, where this insertion sequence is excised from the target from the donor DNA, and it is taken into the target DNA. Okay, so this mechanism is non-replicative. Okay, so these are main three types of transposable elements in bacteria: insertional sequences, okay, composite transposons, for example tn9 and tn10, and you have non-composite transposons, for example tn3, which transposes by co-integration mechanism. This co-integration mechanism help in the formation of HFR strains as well. How? Let me show you. Okay, so suppose this is the plasmid, this is a plasmid here, and it has this transposable element, and suppose this is chromosome. Since we are talking about bacteria, so both will be circular. So this transposable element will help this plasmid, but you must know that HFR forms by F positive plasmid, okay, which is responsible for conjugation. This F plus this sorry, this F plus plasmid will get integrated into the chromosome, and hence it will result in the formation of an HFR this strain. Okay, what is HFR strain? HFR means high you can see recombinant. Okay. Suppose this is the bacteria you have. It has chromosomes and it also has, say, this F plasmid. This is referred to as F plus strain. And the one bacteria in which there is only chromosomes. This is referred to as F negative plasmid. Sorry, F negative bacteria. And this F plus bacteria is able to form conjugation tubes. Okay, so it will form conjugation tube, and this plasmid will then be transferred. Okay, a copy of this plasmid will then be transferred to the F prime F negative, which then changes into F positive. Sometimes what happens? This F positive here, this F positive bacteria, this F plasmid then gets integrated into the chromosome. Gets integrated into the chromosome. Okay, this is referred to as HFR strain. This is referred to as HFR strain. And this integration could happen by these non-composite transposons, as we already saw here. And then this HFR strain will help this bacteria to transfer plasmid plus this tn3. This tn3 into the neighboring bacteria. If this tn3 has antibiotic resistance, so antibiotic resistance, so this will be able to replicate the TN3 into the neighboring bacteria and also transfer antibiotic resistance into that bacteria. So these non-composite transposons are also medically important, as they also help in transmission of antibiotic resistance between between different bacteria. Okay, so I think this is it for today's video. In the next video, we'll be talking about eukaryotic transposons. If you did get anything from this video, kindly consider subscribing, sharing, and liking this video. I hope to see you in the next video. Till then, take care. Bye bye.